you have to do further more too. So before starting with the next two, I want everyone to send the PDF. Is it okay? As you were doing last year, na? So send the PDF by today 6 o'clock. It will be okay.
Yes, ma'am.
Okay
Yes,
for
ma'am.
everyone. All have
Yes, ma'am.
completed the work. Experiments from section A you have done. So send the PDF of that. I'll just see it and then you can move ahead with the next one. And did I tell you all total in that section A? Did we
No,
discuss
ma'am.
all? No, na? How many did we discuss?
Ma'am, only these two, and we were discussing the third, but it was not complete.
Acha, third one is which one? So if you wish na, so I'll tell you today the others also. So
Okay, ma'am.
as I see your two experiments now, I will be commenting that you can move ahead with the next experiment. You can finish up. Today is 15th, children. Okay, set a target that by the end of this month, na, you complete the section A and half of section B. It will be okay for you all or not?
Yes, ma'am.
Yes, ma'am. Yes,
Okay,
ma'am.
as soon you finish the things, it will be good for you only. Okay, करना तो है. Okay, जितना जल्दी खत्म करके रख दो, nicely you make very neat work. Okay, and good file you all make it. Remember whatever you were doing last year, वो सारा कुछ गलतियां मत करना. Very neat labeling in block letters. All pencil work on the drawing side. No pen work on the drawing side. Is it clear? And description will be in this. You'll have to check it in the B part. That whatever diagram you are drawing in front of it, its comments should come. ओके ये सारे नियम बस ध्यान रखना है एंड सेम वे यू डेकोरेट वन पेज फॉर सेक्शन ए नाइसली colorful and then section B which will differentiate the two, okay, separate the two sections, section A and section B, and in front you keep two sheets, one for the section A index and one for the section B index. Both the index you will you'll be writing keeping on top. Is it clear? And above that, one drawing sheet which will be for pasting the certificate. Is it clear? So this way you just do complete everything. Acha, project also you have started, Neha?
Yes,
Neha
ma'am.
and Devanshi? Devanshi, you
Yes,
have
ma'am.
also started. ठीक है देवांशी आपका पूरा का पूरा मिल जाएगा अपने बुक्स में ही. Okay, if you are keeping any refreshers, NCRT पूरा देख लो आप. And then all figures of the NCRT only you will include in your topic, Devanshi. And Neha will have to search in the net also. Okay,
Yes,
because
ma'am.
canopy structure will not get it in the textbook and all. But ecology part, the last chapters, na, they will be related to it. Okay, so now who is opening the lab manual? All have got the lab manual now. Munisha, मिल गई?
Yes. Yes, ma'am. मिल गया.
अच्छा अच्छा okay. Bharti also has. Raj, you also got it. Ritu is not coming. Ritu को क्या प्रॉब्लम हो रही है? रितु एब्सेंट बहुत हो जा रही है. And last year performance was also not good. रितु ने कुछ मेहनत नहीं करी 11th में भी. अब इस साल तो बहुत स्टार्टिंग से करना पड़ेगा. That will not help it if you think of doing it at the end. Okay, you have to understand the things and keep it. प्रिपरेशन बाद में कर सकते हो, उसका कोई दिक्कत नहीं है, बट एक एक चीज को समझ के तो रखना ही है. The things should be very clear so that when you sit for studying, so quickly you can prepare. Okay, that is very important, children. Okay, so now third experiment is which one, Neha?
Ma'am, in this three parts are there. First, collect water from two different water bodies around you and study them for pH. Second
हाँ.
is collect water from two different water bodies around you and study them for clarity and presence of particulate matter in different samples of water. Third is
हाँ.
to study different water samples for the presence of living organisms.
Microbes present in it. Now, what you will be doing, everyone, this will be your third experiment. You write the aim will be to collect the water samples from three different places and study its pH, clarity of water, clarity of water, and
Ma'am,
presence
can you
of.
repeat?
Huh.
from the starting.
Okay, to study the first we have to collect the three different water samples and study. That way you'll write down. To collect the three different water samples and study their pH, clarity of water, presence of particulate matter, presence of particulate matter, and living organisms present or microorganisms present and microorganisms present. So this will be your experiment number 3, aim. Then you can make it as A, B, C as it is given in your lab manual. Okay, so for the pH, so no figure will be there. Is it clear? For the clarity, the particulate matter, you must be seeing one torch picture must be given. Is it there, Neha? Just turn the pages. All of you all can just turn the pages of your lab manual. If it is there
Yes,
in front
ma'am.
of you. Is it given one torch and then they are showing?
Yes, ma'am.
So that figure you will make. And for the microorganisms, now for microorganisms, you will find one full page is there where many microbes are there. Okay, you will not draw all because all we will not be observing in the water samples which you will do the experiment in the lab. You will be getting, so from that now you make the coccus bacteria, mark it there. Could you see first row में देखो क्या क्या है आप read कर दो. So I'll tell that which one you go.
Ma'am,
In
in the
first row,
first, it is
हाँ,
Coccus, Bacillus, Vibrio, and Spirillus.
only coccus you draw. Okay, now second row.
Then second row, Pleurosigma, Navicula, Amphipleura, Triceratium, Nostoc,
हाँ,
Oscillatoria, Spirogyra,
हाँ.
Asterionella,
Wait.
Glutride.
This is wait. This is all in second row.
Yes, ma'am.
Oscillatoria you make it and the spirogyra you make here. Okay,
Yes, ma'am.
done. Now next.
In the third row, Daphnia,
हाँ,
Copod, Rotifer, and Water
so
Strider.
Daphnia you make and copod you make. Okay,
Ma'am,
then
then
now
in the
हाँ,
next row it is amphipod, mosquito larva,
हाँ.
isopods, disticus larva, disticus, and water scorpion.
Okay, only the mosquito larva you make. Was
Okay,
it not?
ma'am.
So
Yes,
total
ma'am.
how many you got?
6.
6. Okay, so these 6 microbes only in this experiment you will be making. Is it okay? All have marked or not? Raj, you got the lab manual or not? Raj,
No, ma'am.
no. अरे तो फिर जल्दी से why you not arranging? What is the problem? Not going anywhere. यहाँ गेल से ले लो. You are not coming to गेल ever.
No, ma'am.
Try to
Going
order
to.
ना. Try to tell your friends जो यहाँ पर रहते उन लोग order कर दे and then you can take up any time. Is it
Yes, ma'am.
possible ना? कि नहीं है possible नहीं हो पा रहा है.
मैम नहीं हो
See,
पा रहा
I
है.
would say children कि जल्दी से अरेंज करके रख लो सारा चीज फिर आपने आराम से घर में बैठ के करते रहना बिकॉज यू डोंट नो व्हेन दिस लॉकडाउन विल स्टार्ट. अभी तो आपको पता है 7 से 12 तो अपने यहाँ शॉप्स खुले ही हैं. यू कैन गेट एवरीथिंग एंड देन लेटर व्हाट विल बी द कंडीशन वी डोंट नो. इसलिए डोंट डिले द थिंग्स. ओके, जितना जल्दी हो सके यू कीप द थिंग्स रेडी विथ यू. All are
Ma'am,
having the pages. All others are having the pages ना.
yes,
बिना
ma'am.
पढ़े the file papers plus your lab manual everything. Okay.
Yes, ma'am. Ma'am,
Devashish
do we
you have
have to write the introduction part which is given before the
no no
experiments?
no no only after the
Okay.
aim whatever is written.
Ma'am, the
Okay.
introduction part is the theory part actually.
The theory is not given there.
Yes, ma'am. After the M, there is no theory, but the introduction is only the theory. It is given. It has given all
Okay,
the things.
but in some experiments so that introduction is too big.
Yes, ma'am.
Shorten it then.
Ma'am, I have
Okay.
written it.
So you have written the whole thing,
Ma'am, I have
Ma'am, it is
also
not
written it.
actually that big.
not that big. In
No,
the
ma'am.
two which you have done. In the
No,
two
ma'am.
which you have done, short only.
Yes, ma'am, it's of an inch, medium length, not too long, not too
Ma'am,
short.
like there are 4 to 5 points of each introduction.
Acha, acha. Okay, so then that much can be written. Okay, because I'm not remembering that part. Some I'm remembering that it's a one full page introduction is there. So don't write that much. If you are doing this water sample one, remember just what is water, composition of the water, and then brief. They'll tell about the clarity. In this, how many lines are there in this experiment, the third one which we are discussing just now?
Ma'am, it has three small paragraphs.
Three paragraphs are there.
Yes, ma'am.
Okay. So what all they are covering? Just tell
Yes,
first paragraph what they are telling.
ma'am, it is saying that the addition of some substances or factor which degrade the quality of water so that it either becomes health hazard or unfit for use is called water pollution. And
Huh.
then most surface waters usually contain small quantities of suspended particles, organic substances, and number of living organisms. When concentration of these substances in water increases, the water becomes polluted and unfit for use. Some other substances pollute water even at
So
low concentration.
this is all first paragraph.
Yes, ma'am.
So it's very big. So what you do, they are telling about the pollution children. Try to write only 3 lines of pollution. Okay?
Yes, ma'am.
Summarize it. Okay? From this whole paragraph related to pollution, just write 3 lines. Okay, next paragraph what they are telling.
The presence of organic waste in water promotes algal growth, resulting in the depletion of oxygen in water. It is called eutrophication.
Huh.
The later leads to that of aquatic organisms.
Okay, so that much you write. Then next paragraph.
Water pollution due to organic waste is measured in terms of biological oxygen demand. It is the
Huh.
amount of dissolved oxygen needed by bacteria to decompose the organic waste present in water. Higher
Huh.
biological oxygen demand of a water indicate presence of microbes in large number due to presence of high concentration of organic matter.
So this much also you write. Okay, from first paragraph, just take it 2, 3 lines. From second and third, you write the whole thing. Okay, this all we will be doing in the environmental issues chapter. Okay, this is all theory part which you have to do. Okay, so that can be done in the theory part.
Ma'am, ma'am,
Huh.
but in
Say,
first two experiments, I have not written the theory. So should I write them in the last of the experiment?
no, now leave it.
Okay, ma'am.
Do not write them. Others, what about other students? Barbie, Shravani, Rumisha, what you have done?
Ma'am, I
Ma'am,
have
I have
written
written
the
the theory and
introduction.
then I have not titled it as introduction, but after aim requirement, then I have added the theory. In theory, I
Huh.
have added the introduction part and then
Uh-huh.
the
Okay.
procedure.
Acha, good, good, good. Okay, Shravani, you also?
Yes, ma'am. After aim, I have written down this theory. Ma'am, in the
Okay, you have
beginning, I have written in the form of introduction only.
again. Yes,
Ma'am, in the
Rumisha, what?
beginning, I have written the introduction part of that
Acha.
experiment.
As it is given in the book, same way you have written.
Yes, ma'am.
Acha, you have not written it as a theory, subheading.
Yes, ma'am.
Okay, okay. So after that introduction, which is given in the lab manual, then you have started with the aim.
Yes, ma'am.
Acha, okay, okay. So now from next practical onwards, don't do that.
Ma'am, actually,
Okay.
for some practical, there are some parts and particular theory is not given, so I have written it as the introduction for whole of the experiments.
Okay. It has to be written combined only, na? Because one, two, three, if the three parts are there, na, for each one you don't have to write theory. Okay, aim one will be the combined aim. Then A, B, C suppose it is there. तो ए का एम अलग हो जाता है, बी का अलग होगा, लेकिन उसके साथ सिर्फ मटीरियल्स रिक्वायर्ड वेरी करेंगे. थियोरी फिर उधर नहीं लिखना है फिर से. इज इट क्लियर टू यू ऑल?
Yes, ma'am.
Do not repeat each part. Okay. So this third experiment is clear to you all? Okay, one common aim, then this theory what I have discussed just now, then you will take up as the separate one. Followed. And if you want to combine it in your own way na, so then that can also be done. Followed. But then you have to organize in a proper way. Okay, materials required, then its procedure will come, then its observation will come. Okay, and if you want to write down aim also separately as in the lab manual they have given, so you can write that way. Okay, now the next experiment they have, fourth one. राज को तो
Study.
होगा, लैब मैनुअल भी नहीं है. तो राज, did you complete the two experiments or not? राज
Yes,
जल्दी
ma'am.
बोलो, आपने दो कर लिए?
Yes, ma'am.
How?
मैम मेरे पास पेजेस थे इसलिए.
नहीं तो वो मिला कहाँ से एक्सपेरिमेंट? नेहा यू सेड.
Yes, ma'am. I have sent it in the group.
अच्छा अच्छा, ओके ओके, तो फिर राज कर पाए. ओके, पेजेस वर देयर. ठीक है ठीक है. ओके, सो नेहा नाउ नेक्स्ट फोर्थ वन.
To study population density and percentage frequency of different plant species of a given area.
Okay. So this is in the book, in the lab manual, they have given separately or one experiment.
Ma'am, one experiment and ma'am, one only.
Only one. In one table only they have written the frequency plus the density. Just check the observation table.
Ma'am, there is only one table and it has everything.
It has everything. So do it the same way. There's no problem in that. Okay, and
Yes, ma'am.
diagram na, you make one quadrant they have shown where they are making just small grasses sort of. Neha, can you show the figure sitting on the camera?
Yes,
Similar
ma'am. I
figure
will.
from there.
Yes,
Where
ma'am.
you
Yes, ma'am.
find the different plants are distributed in that quadrant. मतलब एक square के अंदर.
Yes, ma'am. I got it.
हाँ, that figure only you will make. And in this, the theory is given, na? It's given in the theory.
Ma'am, it is introduction only.
Introduction only.
Yes, ma'am. And that introduction is only the theory.
So the formula and all is given in the
Yes, ma'am.
introduction. Formula is in
Yes, ma'am.
introduction. Okay. So then you just define the density. When you write down the theory from the introduction, you take up what is the population density, its formula, and what is frequency, its formula. Only that much you take down in the theory part. Is it okay?
Yes, ma'am. Yes, ma'am.
Correct. And only one figure which is showing one square inside which the glasses sort of they are shown. बस
Yes,
that
ma'am.
figure you have to make. क्योंकि एक साथ में और भी है जिसमें you find that square pyramid. You do not need that much. Only one
Ma'am,
figure.
we have only one figure only.
One figure only. So
Yes,
that's
ma'am.
okay.
Because others are
Yes, ma'am.
graph.
Graph you don't have to make.
Okay, ma'am.
हाँ. Okay, the graph one you will not make. Okay, now next experiment.
To prepare temporary acetocarmine stained mount of onion root tip to study various stages of mitosis.
Mitosis. So in this experiment, no changes will be there. As it is given, same way you copy. Okay, just one thing you remember that you have to make four phases. There I find that early and late is also given. See the figures.
Yes, ma'am.
Early and late given. You make for all late. Is it clear? One will be prophase, then the metaphase. So metaphase also late one. Okay, then anaphase also the late. And then the telophase also one. Got it now, everyone?
Yes, ma'am.
So the comments are very few children. So but two pages you make it. Okay, two on one page so that the diagram comes proper on that opposite side and then the two on the next page. Is it clear?
Yes, ma'am.
Okay, so this mitosis is over. Now what else?
Ma'am, it has two more diagrams.
Which one?
One it is showing the onion root tip with all the cells. Like there are two diagrams, and one in a bottle there is a onion and water. There's
Yes,
two
ma'am.
diagrams.
That one. Don't make that bottle one. Okay, that all we do it in the lab, but you make only the stages.
Okay, ma'am.
Okay. What we did in the 11th class was cell division.
Yes, ma'am.
So those stages only we made. Don't make that procedure figure where they show the beaker with the onion dipped in it. No?
Yes, ma'am.
That one we did. Done,
Ma'am,
na?
then this. Yes, ma'am.
Huh.
Sixth experiment has again three parts. First
Huh.
is to study the action of salivary amylase on starch. Second,
Huh.
to study the effect of different temperatures on the activity of salivary amylase on starch. Then third, to study the effect of different pH on the activity of salivary amylase on starch.
Now what you do, children? First one, so you will not write. Okay, you will do only to study the effect of temperature and pH on the activity of salivary amylase. Aim will be this one. What will be the aim? To study the effect of temperature and pH on the activity of salivary amylase. On the activity of salivary amylase. Done, na?
Yes, ma'am.
And then everyone understood Bharti, Shravani, Vidisha. Are you all marking?
Yes, ma'am.
Yes,
So
ma'am.
make one common aim. In that you make it as A and B. And the first part which is given that you need not record it in the file. Okay, only two you will record. And for the pH one, now see there, for the pH they must be giving you one beaker with the test tubes kept in it,
Yes,
where
ma'am.
37 degrees Celsius is written. It's there, one
Yes,
beaker
ma'am.
with three different test tubes are there.
Yes,
So
ma'am.
that you have for pH. Okay, and now see the temperature one. So in the temperature one, you find that three beakers with the tripod stand you will find. Okay, and one below the other they are. You have that way.
Yes, ma'am.
And side now you find the test tube stand is there with the different test tubes. It's there.
Yes, ma'am.
So do not make those test tube stands. You will make only the tripod stand with the beaker. So three different temperatures must be there. Plus minus 5 is there.
Yes, ma'am. Yes, ma'am.
So that setup you make. Okay, उसके आगे भी देखना तो one test tube stand with the test tube series is there. तो वो नहीं बनाना. Okay, then second one again a tripod stand must be there with the spirit lamp and then the beaker where it is showing 37 degrees plus minus. Is it?
Yes, ma'am.
And the third one is your what temperature? Plus
80 degrees
minus.
Celsius.
Okay, so this way. So you will make only the beakers. Done. All have marked Barbie, Shravani, Devanshi.
Yes, ma'am.
Yes,
Devanshi
ma'am.
understood. Clear na Devanshi?
Yes, ma'am.
वो टेस्ट ट्यूब स्टैंड तीनों में से कोई भी मत बनाना. You make only the beakers with the test tube in it. Okay, वो सीरीज पूरा टेस्ट ट्यूब स्टैंड
Yes,
वाला
ma'am.
do not make children. Clear na?
Yes,
So
ma'am.
now over section A.
Ma'am, one last is left.
DNA isolation is also there na?
Yes, ma'am.
DNA isolation.
Yes, ma'am. That is there.
Last one. Read it out.
To isolate DNA from available plant material such as spinach leaves, green pea seeds, papaya, etc.
Huh. For this experiment, no change will be there. You copy the whole thing. And one diagram is given. That diagram only you have to do. Okay?
Yes, yes,
And
ma'am.
just theory part, only few lines you take up from the introduction if it's there. Only 4 or 5 lines, just what is DNA, that's all. Okay, don't elaborate.
Yes, ma'am.
Done everyone?
Yes, ma'am.
Yes, ma'am.
So I hope everyone understood and what is the date fixed for completing this much?
till
What
last of
date?
April,
So date you say na? What date?
30th April.
30th April. Okay, Raj हो जाएगा.
Yes, ma'am.
हाँ ठीक है, 30th April तक खत्म कर दो. ये फाइल का काम तो यू कंटिन्यू डूइंग एट योर ओन लेवल. ओके, वेन एवर यू गेट द टाइम, कुछ पढ़ने का मन नहीं करता है, टीवी देखते देखते भी ये काम खत्म कर लो. Okay, just that way you can finish up this work. Okay, but before that, na, today you send me the PDF of the two experiments. Achha, one more thing now. Two students have got the project. Others have selected the topic or not for the project.
Yes, ma'am.
Yes, ma'am.
Rubish, to say quickly.
Study the effect of antibiotics in on microorganisms.
Okay, good. You can do that. Then Shravani,
Ma'am, study of apical dominance in potato and colors.
apical dominance. Don't take apical dominance. Okay,
Okay,
that
ma'am.
will be the 11th class one. Okay, you have to select the topic which is related to your theory. Okay, this year's syllabus. Okay, antibiotics so we will be doing in the this unit only which we are doing. So Rumisha can take down. Barbie you have selected.
Yes, ma'am.
What did you select?
Ma'am, actually, I have selected the frog, the ovary during the breeding season. I really
Uh-huh.
like this, but
Animals.
I'm
Uh-huh.
but I'm not sure because as
Uh-huh.
the cockroach
Uh,
stayed in 11,
see,
that's why.
I will suggest don't take it because nowadays na, it's very strict that you cannot catch any animal and you cannot kill it, you cannot do any experiment on the. So one day principals have also said that do not do it. Otherwise, so we were doing. Okay, but
That's
it's
why,
so
ma'am, I have consulted with you.
because now since it is banned, so we have to stick to it.
Okay.
Okay, so we will not do that. The students have been doing this experiment and they have been opening the frog and studying very nicely the ovaries, and you can see very nicely the eggs also children. Okay, उसको मैचरेट करके ओवरी को देख पाते थे अच्छे से, but now we will not prefer this experiment. So Barbie, you can think of something else.
Ma'am, then the study of morphology of the frog's tadpole through the specimen.
Acha, life cycle of the tadpole. Okay, think of something else.
Okay, ma'am. Then
Ma'am,
That study will not be related to your theory part. It will be just wasting the time. अब बस देवांशी has selected ना that topic, तो देवांशी will be able to cover up the whole of the one chapter. तो वो उनकी पूरा preparation हो जाएगा बोर्ड के लिए. तो select such.
Ma'am,
Okay.
then the drug one.
Drugs one.
Drug
हाँ okay.
resistance
हाँ
in
you
the bacteria
can
using the antibiotic.
which one? Which one?
Ma'am, the study of drug resistance in bacteria using antibiotics.
Acha. And Rubisha, what you are taking?
Study the effect of antibiotics on microorganisms.
Okay, so both will be quite same. Barbie, it will be same. So procedure will be same and then almost same thing we will be covering up in this Rumisha's only. Okay, culture medium you have to prepare, Rumisha. Okay, in the lab and then you'll grow it. Grow the microbes. You'll culture the microbes and you'll study the antibiotics effect. Okay, and same as Barbie yours also, that's what you are saying. So by the time see Shravani could select something.
ma'am, can I do this that comparative study of the chlorophyll content in 5 different species of plant?
That is also last year's, no? That also leave it. Okay. It will not be related to your theory, na? So do not take such topics which will not be
Ma'am, then
useful.
to study pollination in sunflower and legume.
Acha, pollination. Okay, so what work will be done? That you read it first. Procedure part. That what experiment you will do it in the lab or what investigatory. Because the project, whatever you will be choosing, that will be investigatory project. Okay, not just seeing the net and searching the things and then writing. Okay, as Neha is doing the canopy, so I said that you visit, okay, all around and try to study, try to take the pics of the different canopies. Okay, so that has to be done. And then try to observe how the temperature varies. Okay, under the trees, different trees, and in the open area, in the ground. Okay, that all. So some work you should do. हाँ श्रावणी, just see by the time देवांशी का हो गया, राज आपने कुछ सिलेक्ट किया क्या?
No, ma'am.
नथिंग. अच्छा क्योंकि बुक रही हैं. ठीक है तो Rumisha can take down the synopsis quickly and others can think about it. And see, one thing can be done, children, as you select the topic now, you can make a call and I'll give you the synopsis that way. Is it okay? Yes
Yes,
or
ma'am.
no? हाँ, डू नॉट वेट फॉर द क्लास. अगर डिसाइड हो जाता है, आपको समझ में आ जाता है कि दिस आई वुड लाइक टू डू, तो यू कैन मेक अ कॉल and then I'll give you the synopsis and you can start your work. Okay, otherwise then the class time will go. Okay, it will not take much time. So Rumisha, take down quickly the synopsis. First will be the introduction. Noting, Rumisha.
Yes, ma'am.
So first will be introduction. Then next we will have the microorganisms. Then take up microbes. So you write about the useful microbes and then the harmful. In this microorganisms topic only, try to mention there the useful microbes and the harmful. So harmful ones will be then the pathogens. Okay, so you will have to write in brief about the useful, then you will take up the harmful. In the harmful, again you take up the different categories. Okay, that is the bacteria, then the fungi, some viruses. Okay, and then what diseases they cause. Then you take up in detail the bacteria. Okay, fungi viruses को बस brief में खत्म करना. Okay, bacteria, harmful bacteria and the diseases which they cause, that you take up in detail.
Okay, ma'am.
You wish understanding, na?
Yes, ma'am.
Okay. Then after completing that, what are those bacteria and what are the common bacterial diseases? So try to cover up those bacterial diseases which we have done in theory. Is it okay, Rumisha?
Yes, ma'am.
That try to study in detail. Okay, then you take up the prevention. Or do not take it as prevention, you take up now direct antibiotics. Okay, antibiotics. So in this topic, you write down what are antibiotics, how are they made, okay, how are they made, their mode of action, done.
Yes, ma'am.
Okay, then after that, common antibiotics used, common antibiotics used. Okay, and then you will mention that these are the antibiotics against which bacteria, or for which disease the different antibiotics are taken. So you will try to study, you will open the net, search in the net, and then you will get a different sites, try to visit and try to take down such things, Rumisha, which you understand. Okay?
Yes, ma'am.
क्योंकि आपको बहुत सारा मिलेगा जो कि बिल्कुल बहुत ज़्यादा डीप है, तो उतना नहीं. जो आप समझ पाए and you are able to explain in the class.
Yes,
Okay,
ma'am.
after you have done the experiment, you will be explaining in the class. What are antibiotics? How are they made? How do they work? Okay, the different antibiotics will have the different mode of action. इसके बारे में थोड़ा सा पढ़ना. Is it clear?
Yes, ma'am.
Okay, then the last will be your, and then same the aim materials required will start. Done, na?
Yes, ma'am.
Okay, so the harmful effects of, that also you write down. The side effects of antibiotics. Side effects of antibiotics. That you take it as last.
Yes, ma'am.
Done, na?
Yes,
Okay.
ma'am.
Now procedure and all, तो when you do the experiment that time only I'll be telling you. Okay, जब लैब में करेंगे तो अपना तो 41 तो ऐसे ही हो गया. So today we could not start with the immunity one. So we'll end up today the class, children, and other students, those who have not selected the topic, they will be selecting the topic and they can make a call and take down the